Bismillah ar-Rahman ar-Rahim. So I welcome you all in this lecture. Today we are going to discuss about the role of Wnt and GPCR signaling pathway. And before we begin, we should like to know how Wnt signaling pathway is different from RAS and RAF signaling pathway. So Wnt is basically a ligand. Upon its activation, in the absence of Wnt signals, there is an active compound which is called as GSK3 beta. Now this GSK3 beta is basically meant for the phosphorylation of numerous proteins. These phosphorylation of these numerous proteins like APC, beta catenin leads to an early degradation of these proteins. Now these proteins being phosphorylated by GSK3 beta are unable to translocate inside the nucleus. That means they are unable to proceed the cell towards proliferation or differentiation and motility. So the cell is unable to proliferate or differentiate further in the absence of these signals, right? And motility. Alright, so once the Wnt signals are present, that GSK3 beta became phosphorylated. Now this brown color is being phosphorylated. This leads to an activation of GSK3 beta and the release of APC and beta catenin. Now beta catenin and APC are unphosphorylated. They are not phosphorylated, so that leads to the translocation of beta catenin from cytoplasm inside the nucleus. Now, once they are moved inside the nucleus, they attach themselves to the DNA, and from DNA binding, they are responsible for triggering all these three mechanisms: proliferation, differentiation, and motility. So, Wnt signaling, so far we have known that in Wnt signaling, 19 Wnt members have so far been identified in different human tissues. Half-life of beta-catenin increased from 20 minutes to 1 and 2 hours in Wnt activation. Additionally, it has been known that apart from beta-catenin, which is being placed inside a Wnt signaling pathway, beta-catenin protein are also localized in E-cadherin junctions. Now, so far we know that the cell-to-cell interaction, consider this as cell A and the neighboring cell as cell B. Now there is a junction which is called as adherent junctions. In adherent junctions, the binding affinity, the binding of adherent junctions is maintained by E-cadherin, being E as the name indicates, E-cadherin, E-cadherins. Now, E-cadherins are basically representing the cells which are epithelial in origin. Now, it is known that these E-cadherin molecules, which are replaced over here as in red color, I am just intentionally making them in red. Now, these adherent dimers are actually attached inside the cytoplasm via B-catenin. Okay, so loss of B-catenin also results in the loss of connectivity of two cells staying together. Wnt are powerful morphogens in mitogens. The role of beta-catenin in tight junctions has also been established. When E-catenin expression reduced, beta-catenin is shifted to the nucleus for transcription activation of other factors. However, there is an interesting, what you can say, the puzzle is still remains, that inside the cell there are two forms of beta-catenin present. One form in which APC, beta-catenin are present inside the cytoplasm. In an alternate form, inside the adherent junctions, they also remain bound to this adherent junction, and generally we can target them in E-cadherins. Now the puzzle is that beta-catenin present in adherent junction is the same, but it is present in a different form as compared to the one which is present inside the signaling pathway. So the scientists are still deciphering the reason why there are two alternate forms of beta-catenin present inside the cell. That's the puzzle. It is known that in breast cancer, the Wnt signaling activation is 4 to 10 times higher as compared to the normal cells. Similarly, in prostate cancer, 5 to 10% mutated forms of beta catenin are observed. This mutated form of beta catenin is unable to be phosphorylated. Once the beta catenin is unable to be phosphorylated from GSK3 beta, this means this beta catenin is unable to be degraded and its active form remains active inside the cell for a persistent proliferation and growth. Now, these mutated forms are also reported in other different cancers: colon, liver, ovarian, and endometrium cancers. This is all from Wnt signaling pathway. Now, the other pathway we are going to rush through is GPCR pathway. Now, this is a cell membrane, and GPCR, as the name indicates, these are G protein coupled receptors. Now, G protein coupled receptor is basically a long receptor which is actually transferring inside the cell membrane in seven ways, in seven types. Now count them: 1, 2, 3, 4, 5, 6, and 7. Now this is G protein coupled receptor. G protein coupled receptor. Now in this signaling pathway, this G protein signaling receptor is basically hosting any signal from the outside source, and upon the introduction of any signal from here, this GPCR coupled receptor is responsible for the activation of what you can say a trimeric complex. Let me draw 3 different colors over here so that we can get a very good understanding about what we are looking at. Okay, so let me change the color for because it's again confusing. So these are the G trimeric proteins I am labeling right now, and let's use blue color, and this is alpha, G protein alpha, G protein beta, and G protein gamma. Okay, so once there is a ligand bind with the GPCR receptor, it leads to the activation of this trimeric complex, which is called as labeled as G alpha, beta, and gamma. G alpha has a binding affinity with both GDP and GTP. Obviously, in its inactive form, G alpha remain bound to GDP. But once it bound with G alpha bounds with GTP, it is mainly responsible for almost similarly the same mechanism as we have noticed in RAS. So it is engaged in PIP pathway. It is also engaged in ARAC pathway indirectly. So the root of G alpha GTP activation is also mediating for the same proteins complex and is indirectly activating the same signaling mechanisms. Alright, the next thing about this GPCR pathway is elaborated over here. Now there is a ligand, then there is a 7 transmembrane protein structure, and if we focus over here, G alpha and beta and gamma remain in active form in GDP. Once it is active, it is responsible for the both stimulatory and inhibitory role. Now, from stimulatory role, it is responsible for the activation of cyclic AMP via adenyl cyclase enzyme, and then it is also responsible for phospholipase C beta activation, which leads to the cleavage of PIP into inositol and diacylglycerol. So PIP is converted into PIP3 is converted into IP3 and diacylglycerol. Remember the RAS signaling pathway in which the two signaling pathways we have discussed. So the phosphorylation of the lipids is being induced by this RAS, and over here the phospholipase C is being influenced by GTP and alpha G protein complex. The second one, there is an inhibitory role also being noticed in GTP, G alpha protein inhibitory role. It leads to the inhibition of cyclic AMP as well. So these are the roles which are being conduced by GPCR. It depends upon what type of signal is being transmitted to the cell. Interestingly, it has also been noticed that this G protein coupled signaling pathway also is responsible for what you can say a momentary inhibition of certain stimulatory factors, and this inhibitory response is being noticed inside the eyes, rhodopsin. So, second mode of signaling by GPCR is also been deciphered. So, earlier we have talked about no receptor, there are no ligand present. Now there is a ligand present, so it's somewhat similar like wind signaling, no wind signaling, and if there is a wind signal present outside. But the opposite role, inhibitory role and stimulatory role of GPCR makes them unique. Now we are talking about that depending upon the ligand, they may are responsible for a sort of inhibition. So that inhibitory second mode of signaling by GPCR is first deciphered in rhodopsins. Upon photon ligand exposure, signal activation of GPCR is halted by arrestin, a negative feedback. Two paralogs of retinal arrestin, termed beta arrestin 1 and 2, have been found to be expressed in virtually all non-retinal cell types throughout the body. Now they have been observed in all non-retinal cell types, but earlier they were identified in retina. So upon the exposure of any photon energy emission, the retina GPCR signaling tries to calculate and momentarily halt the stimulation. And similarly, the GPCR is also engaged in small cell lung carcinoma. How? The cigarette smoking led tumors contain peptides. Now, these peptides are called neuropeptides. Examples of these neuropeptides are bombesin, gastrin releasing peptides, bradykinins, cholecytokines, gastrin, neurotensins, vasoprins. And now, these vasoprins and other receptors lead to the same activation of small cell lung cancer in this way. So instead of ligand, if we say there is a release of bradykinins, vasoprins in small cell lung cancer, we can observe the same phenomena. And this small cell lung cancer is being observed in smokers. So a prolonged exposure of smoking leads to the release of these ligands, and these ligands are responsible for the activation of carcinogenesis in an early phase. Thank you. So far, that's what we have deciphered so far about the wind and GPCR signaling. Keep watching my channel. Thank you very much for your valued time and attention.